talking about a process which occurs at the inner mitochondrial membrane called oxidative phosphorylation. So here's the inner mitochondrial membrane. Start with glucose out there, and we convert glucose to what? Pyruvate. Pyruvate is going to come into the mitochondrial matrix, and it's converted to what? Acetyl-CoA. You guys, when you draw this stuff yourselves, it doesn't have to be beautiful and pretty. I know what G means, I know what P means, I know what AC-CoA means. As long as you're comfortable and you have the other information, this is fine. So, that's going to get oxidized through the citric acid cycle. And overall, is what we've done is we've produced a whole ton of this molecule NADH. It carries 2 electrons. They're very high energy electrons, so this has a high G value. Okay, I need to get the energy from there into ATP. Okay, so my challenge is to get the energy from here, this molecule, into that one at the end. Okay, so as what we have is a system that involves 5 proteins. I'll start off with 4. Okay, so there are 4 proteins that live. Now when I say there are 4 proteins, guys, I don't mean there are only 4 proteins. There are 4 types that we're interested in. So think about this in 3D, all the way around the mitochondria. Complex 1. Then there's a protein called complex 2. Guys, complex 3. 4 here. And the one over here is called ATP synthase. So all these names are in your books, so don't worry about writing them down. You know the names. Okay, so what's going to happen is the electrons are going to move through this chain of proteins. So the electrons come from here, and they go into complex 1. What has happened to complex 1? What just happened to it?
It's been reduced.
Got reduced, exactly, yeah. NADH got oxidized, and we got NAD+. That goes back out, it is remade into NADH. But the electrons are going to make a series of jumps through these proteins. As they jump, they lose energy. So if we think about the energy of the electrons, G, and this is 1, 2, 3, 4. As the electrons move from 1 to 2 to 3 to 4, they're going to lose a little bit of energy. So they come into 2. So they're lost from here, so they're oxidized. As the electrons jump from NADH into complex 1 and onto complex 2, energy is released to move to pump protons. Electrons are in complex 2, then they get sent to 3. When they jump into 3, energy is released and protons are moved across here. There's a whole pool of protons down here, and they get pushed across the membrane through these pumps. The electrons jump again, and protons move. Okay, so that's what we've done. We've taken protons from down here, and we're pumping them into this space here. So I've got a concentration gradient of protons. So if you think about the concentration gradient of protons, lots of protons, not so many protons. Which way are they going to flow? Are they going to flow down or up that gradient? Down, if they could. Can they do that across the membrane? They can't just move freely across the membrane. They can move down this thing here though. So this acts like a channel. So the protons come back down through here. So you've got a store of protons up here. They just flow back down. And then more NADH comes in and they get pumped back again. So it's constantly flow down and pump back, flow down and pump back. As that happens, the energy up here is released and used to make ADP into ATP. So this has a low G, this has a high G. Energy to go from low to high comes from this gradient collapsing. So it's an example of something I said earlier, of coupling different processes. The electrons get here. When there are 2 electrons here, you can't put any more in over here. It's like a roadblock. So what we do is we take some oxygen at complex 4, we add the electrons, we add some protons, that makes water. Electrons, oxygen and protons make water. That's where you need your oxygen, that's where the water is released. So as that happens, you oxidize this, you reduce the oxygen and you make water. Make sense? It's tiny.